make yourselves comfortable. Again, thank you all for coming to Grand Rounds, and we're really privileged to have two exceptional talks from talented researchers looking at, I think, really novel approaches to understanding cancer biology and really fundamentally our understanding of areas that we don't necessarily think about every day in the course of cancer therapy and research. And we'll start first with Dr. Manoj Pillai. Manoj, as you know, is an associate professor of medicine in the section of hematology and a very productive physician scientist whose interest has included regulation of hematopoiesis and as particularly an interest in RNA mechanisms and splicing, which I think increasingly we understand is driving not only myeloid malignancies, but probably other diseases, other malignancies in a very fundamental way. Manoj received his medical degree at All India Institute of Medical Sciences in New Delhi, completed his fellowship at the Fred Hutchinson Cancer Research Center, and now, of course, is a valued senior member of our faculty and here to teach us about this. So Manoj, thank you.
Thanks, Charlie, and for the invitation and the introduction. So as Charlie said, I'm a physician scientist. I attend on the bone marrow transplant floor for the most part. In the lab, we study hematopoiesis and mechanisms of aberrant hematopoiesis. So today I'll touch base on how these group of splicing factor mutations have kind of changed our understanding of how they can function as oncogenes when mutated, and how we are still in search of a mechanism and trying to therapeutically target it. So the disease that hematopoietic biologists are often interested in, and I'm no exception, is myelodysplastic syndrome. And this is a disease that attracts us because it manifests in hematopoietic failure in its most myriad forms. And it's defined by this trifecta of ineffective hematopoiesis, mutations, and genomic instability that leads to clonal expansion. And it's not that rare a disease, but considered clonal myeloid, although it's a hematopoietic stem cell derived disease, and it's a disease of the elderly, mostly older than 70. Epidemiological causes are still being worked out. Toxin or radiation exposures have been linked. They are clinically very heterogeneous, so they have a variable clinical course that ranges from indolent to rapidly progressive leukemia. Bone marrow failure manifests as anemia, thrombocytopenia, or bleeding, and infection. And the bigger risk is, of course, that a third of them, due to the genomic instability, will transform to acute myeloid leukemia, often called secondary, or TAML, which are difficult to treat and do not respond to standard chemo. In the pre-genomic era, we had distinct clinical syndromes like the 5q syndrome or the CMML and the RARS, which are probably less relevant now since we know more about its genetic basis. And as I mentioned, next generation sequencing has really equipped us with more of knowledge of its genetic background. And I've just put some pretty pictures out here that shows myeloid dysplasia here. These are erythroid precursors that have iron laden due to mitochondria getting laden with iron. And this is a dysplastic megakaryocyte, which is a precursor of platelets. So MDS of late is seen as a part of a spectrum of clonal myeloid disorders. So normal hematopoietic stem cells can often lead to what is called clonal hematopoiesis of indeterminate potential, in which you can detect clones in normal individuals, often older ones, but they do not really have a hematologic manifestation, so their counts are normal. And then, as illustrated here, further along in the clonal process, there is MDS, which obviously has clinical manifestations. And then when you have a proliferative process, then it is AML. So there is clearly a spectrum. And the reason why we think it is a spectrum is also because the mutations in these disorders are often shared, although not identical. So in CHIP, there is a preponderance of epigenetic regulator mutations. In MDS, it's probably half and half of epigenetics and splicing. And in AML, you do have more of other mutations like those of FLT3 or NPM1. So there is clearly a component of aging and genomic instability that plays into MDS pathogenesis. The mutations and karyotypic anomalies of MDS have been worked out in the last few years with whole exome and whole genome sequencing. And what we know is that about half or more of patients have mutations in splicing factors. About another half have mutations in epigenetic regulators. And there is considerable overlap. About a quarter of them have mutations in both of them. And the rest of them have a smattering of different mutations in TP53 or other transcription factors and signaling pathways, etc. The focus of the talk being splicing factors, and that's our interest. We'll focus on that today. So RNA splicing is an enormously complex process with many different players, and Yale has a proud history of contributing to much of its understanding through Joanne Stites and other investigators. So the cartoon on the left is obviously just to demonstrate back in 2011. This is even before all the cryo-EM structures were available. There are more than 300 proteins that are organized into 5 small nucleoprotein particles, U1 through U6. And there are multiple steps of catalytic activation, and that results in the removal of the intron and the joining of the 2 exons. I've just demonstrated it in a little more simplistic fashion here. The 2 exons, the intron that have the 5' splice site, the 3' splice site, the branch point, which is typically an adenosine, and then this polypyrimidine tract. And then in sequential steps, there are 2 critical transesterification reactions in which there is a production of this lariat-like sequence, and then the second step, the 3' end of this exon will do another nucleophilic attack, and the lariat is removed and it is joined. So very complex, very conserved, and obviously has a lot of relevance to disease. Now, you think of splicing, biochemists talk about constitutive splicing and alternative splicing. Constitutive splicing is when the exons are always present, you don't remove any of the exons, and the intron is always removed. But if you have competing splice sites, the 5' or the 3' splice sites, you can have variations of the theme where you have alternative 5' selection, alternative 3' splice site selection, or removal of an exon, that's a cassette exon. The whole intron can be retained, or you can have mutually exclusive exons. This is just how alternative splicing is characterized, especially in next generation sequencing analysis-based data sets. So splicing aberrations are now known to be widely prevalent in cancer, and the most comprehensive study probably is from Gunnar Reich's group that came out in 2018, in which they looked at 32 cancer types across the TCGA, more than 8,000 samples. And what they found was that all cancers typically have about 30% more of alternative splicing events. And they have around 930 new splice junctions. A new splice junction is when a new exon-exon boundary is detected with high confidence. But interestingly, not all splicing aberrations are linked to splicing factor mutations. So you can actually have multiple mechanisms that can lead to aberrant splicing. And that's demonstrated here, in which obviously I'm going to talk more about mutant splicing factors as the cause of splicing aberrations, where you can have a mutant factor that leads to a new event, but you can have an increase or a decrease of the absolute levels of a splicing factor. This is more described by Adrian Trainer's group in breast cancer, where you have more of, say, HNRNPs or SRSF proteins that can lead to an alternative splicing event. You can also have increased transcriptions, say like in MYC-driven cancers. You can have too much of transcript, and then splicing itself becomes a bottleneck that can lead to aberrant splicing events. And then you can have cis mutations in the exons itself, which can actually lead to a splicing aberration in that particular gene. So Charlie mentioned the splicing factor mutations initially came to a focus in hematologic malignancies, but it turns out that it is not restricted to MDS, AML, or CLL. So hematopoietic malignancies are indeed the bigger focus, but you do have a touch of these in some of these cancers, like mucosal melanoma, uveal melanoma, and some in breast cancers, lung adenocarcinoma. Over the more than 300 splicing factor proteins, only about 8 of them are known to be mutated, and of which 4 are known to be mutated in high frequencies. And interestingly, they are mutually exclusive. So you don't have mutations in each of these factors that occur along with the other ones. they're typically non-synonymous, meaning they change the amino acid composition, and except for CRSR2, which is thought to be loss of function mutation. They are associated with specific tissue types and morphology, and SF3B1 mutations occur with ring sideroblast morphology and SRSF2, along with this variant of MDS called CMML, or chronic myelomonocytic leukemia. They have specific associations, although not very tight, like with other oncogenes like SRSF2 and IDH2 mutations, or SF3B1 and TET1 mutations. And we don't really know the biological or prognostic significance of these for the large part. So SF3B1 is what our group has been studying. It is a rather large protein, 150 kilodalton. It's considered to be a component of the U2 complex shown here. So SF3B1 sits here, and it recognizes or stabilizes the interaction of the U2 with the branch point adenosine here. It also, being a large protein with multiple domains, it interacts with multiple other splicing factors like the SRSF1 and 2 that's shown here. It has 20 heat domains. The crystal structure was solved by Lerman's group in 2016, and it shows that it probably makes contact with the mRNA in a second place when it hooks over the heat domains that is shown here. And then the mutations that have been described are all in the heat domains, almost all of them in the heat domains 5 through 9, shown here in these color code. And these, again, we really don't know why these mutations are linked with specific diseases, or what is the prognostic significance of each of them. So in MDS, myelodysplastic syndrome, there is a tight association with SF3B1, and about a quarter, 20 to 25% of all MDS is now known to be associated with SF3B1 mutations, and a fewer proportion in CLL or uveal melanoma. And they are associated with this morphologic feature feature called ring sideroblasts, which are maturing erythroblasts, which are dysplastic, but for some reason end up accumulating iron in the ferric form, and that they can be stained with Prussian blue, and they appear as rings. And the presence of these are not diagnostic of SF3B1 mutations. You can have genetic and acquired causes of ring sideroblasts, but over almost 80% of all SF3B1 mutant MDS do end up showing some form of ring sideroblasts. So when the mutations were initially described, obviously they were just descriptive. We really didn't know, and we still don't know for the large part what exactly is the link between splicing factor mutations and cancer. And so the important questions that were there, some of them which have been answered since, is, are splicing factor mutations founder mutations? And for the large part, we think they are, because of the variable allele frequency of the cells in progenitor populations, and at least some of the mouse models in which you can just have one mutation and you can actually end up with leukemia or MDS. What splicing aberrations result from mutation? And this is probably the question that we have answered comprehensively using next generation sequencing based strategies. The important question that remains unanswered is, how do these mutations lead to clonal dominance in specific phenotypes? And the big question being, is aberrant splicing directly responsible, or if the mechanism of oncogenesis is unrelated to splicing, but something that is distinct from a splicing outcome? And those include multiple mechanisms that folks have been working on, including R loops, or it's the interaction of these factors with chromatin, which is a focus of my lab's efforts, and if RNA-mediated epigenetic changes, which have been described again, are they in part to blame? And for clinicians and for therapy, there has also been a question of whether splicing factors mutations can be directly targeted therapeutically, and I'll come to that. Unfortunately, the answer so far seems to be no. So these factors have been really challenging to study their biology, and for multiple reasons. One is that they are single allele mutations, so if you overexpress them in cell lines or model cell systems, there is the obvious criticism that they are not really representing physiology. The native sequence of some of these are actually toxic to E. coli, and you cannot even clone them. So we had to actually, for SF3B1, we had to change the sequence by codon optimization algorithm so that E. coli will allow its propagation. The cell lines, for some reason, hate these and rapidly mutate or silence these, especially on prolonged culture. And this is a dirty secret within the field that it's virtually impossible to maintain these cell systems for extended periods of time. And the mouse models have been kind of disappointing because most of them don't have a phenotype that really reflects physiology. And this is a review from Carl Walkley at St. Vincent's in Melbourne, who has cataloged the mutations. There are 3 mice for U2F1, 2 for SF3B1, and 2 for SRSF2. And there is a CRSR2 mouse, which has not yet been published, but presented at forums. Only one mouse, the Carl Walkley's own mouse, for SRSF2 actually had a disease physiology, disease phenotype as in clonal evolution and an MDS/AML phenotype. All the rest actually had a competitive disadvantage when transplanted. So there are reasons to explain that, but still, these make them difficult to use as accurate disease models. So the approach we have taken for our studies is to either overexpress them in an inducible fashion using a single cistron lentiviral vector, and you can titrate the doxycycline dose so that the amount of SF3B1, the total amount of SF3B1, does not exceed the one in wild type or uninduced cells by more than 2 or 3-fold. The other approach we have taken is obviously to use genome editing. Genome editing of cancer cell lines has proven to be really difficult, and these cells really either silence or lose that allele. But for some reason, embryonic stem cells, murine embryonic stem cells, have been more permissive to these mutations. So we have used a combination of CRISPR-Cas9 and adeno-associated virus editing to actually generate murine embryonic stem cells that are mutant for these cells. And so we have used these 2 models as complementary systems to study the biochemistry of aberrant splicing. So early on, everybody thought naively that all we needed to do was to just sequence the patient samples, and there will be specific genes that will jump out, and those will be the oncogenic answers that we need. Obviously, it turns out it's not as simple, but we also did patient samples and also these induced and uninduced models, and then the RNA-seq at fairly high depth, and then aligned it to the genome. We and others, several other groups, have also published on how the primary splicing defect seems to be one where you have an alternate 3' splice site selection, and this have been alternatively called cryptic or novel 3' splice sites. And we could find around, in the hundreds of novel splice sites in both MDS, CLL, K562 samples. And this is similar to what others have also found analyzing patient samples. And just to summarize what we found, these splice sites are clustered mostly about 15 to 30 base pairs upstream or 5' of their canonical. So this is the new splice site, and this is the canonical old splice site. And so they are clustered around there. The primary sequence is that when we analyzed, we did not really get much of an idea whether there was some sequence specificity that was driving these splice sites to be recognized by the mutant protein and not by the wild type protein. And so in one of our earlier publications, what we did was to actually look at alternative hypotheses as to how these cryptic 3' splice sites end up being selected in the mutant cells. So the prevailing hypothesis of 3' splice acceptor selection is that the spliceosome starts from the branch point and then scans from 5' to 3'. And if it is too close, the AG, which is acceptor site, then you can't really use it as an acceptor. But anything that is suitable in the middle, or it'll use the first splice site that is suitable. And so we looked at maybe beyond the secondary structures, tertiary or primary structure, primary sequence, secondary or tertiary structures might be relevant in this, thinking that maybe if these splice sites are buried within secondary structures, they somehow render them invisible or inaccessible to the mutants, to the spliceosome. So we looked at a few examples in which the cryptic splice site did seem to be buried within, secondary structures within the RNA secondary structures. So we looked at genome-wide in MDS, K562, and the CLL, and what we found was that significantly, almost, in all the data sets, the cryptic splice sites were indeed buried within secondary structures. And we used a series of mini-gene assays that I'm not going to go into for sake of time, which also showed that if you bury a splice site within secondary structure, it is seen only by the mutant and not by the wild type. But if you actually abrogate the secondary structure by taking away the left side of the hairpin, this site will start to be seen by both the wild type and the mutant proteins. So one of the models that we came up is that, at least for the sake of 3' splice site selection, one of the reasons might be that the mutant protein is somehow able to either abrogate the secondary structure or able to use these buried or cryptic splice sites, which is not possible for the wild type. Our analysis and from others, it has emerged that the primary splicing defect is indeed a novel 3' splice site selection for SF3B1, and only a few hundred novel splice sites could be seen such in across the genome, and the change in absolute splicing is rather modest. So you don't see like wide changes in splicing, and the change in the splicing is also cell context dependent. So the CLL splice genes are not the same as the MDS splice genes or the uveal melanoma misspliced genes. To summarize, similar efforts from multiple other groups, including Stephanie has worked extensively on SRSF2 and of late on U2F1. And so her publications also reflect the fact that SRSF2 has a different mechanism. There's an exon selection that will favor CCNG motifs instead of GGNG motif within exons. For U2F1, it is a change in the exon selection because of a -3 position from the 3' splice site, and SF3B1 I mentioned about novel 3' splice sites, and CRSR2 mutants tend to retain U2 introns. So the splicing differences seem to be really different for each of these splicing factors, and there are no common genes or common splice events that seem to be conserved across these multiple factors. We also did some more biochemical work to determine what exactly is the mechanism of aberrant 3' splice site selection used within mutant SF3B1. And for biochemists, the question of SF3B1 was that since it is a component of U2, does the branch point really change, or is it? So the two competing hypotheses that for mutant SF3B1, does it actually change in which branch point they are actually selecting, or the same branch point is being selected, but for some reason the mutant is able to recognize the cryptic splice site because of a reduction in the steric distance that is usually not possible for wild type. And so, but it is really difficult to determine branch point use accurately in vertebrate genomes because vertebrate branch points are degenerate, so there is no sequence that we can really predict. The 3' splice sites are only a few hundred, as I mentioned, so informatic analysis and machine learning algorithms are not really useful. The RNA sequencing does not capture this region because these are introns and spliced out, so RNA-seq datasets actually do not capture this part. And the branch points actually may be redundant, so multiple branch points may exist for each 3' splice sites. So one approach we took was to use unbiased approach based on something that Jay Shendure and George Seelig did in Seattle, where they used a mini-gene library, which does not have the constraints of the genome and the genomic evolution. And what they did was they placed a splice site that is within two degenerate regions here, 25 base pairs each. So you have, analogously, this could be a cryptic, and this is a canonical, and this is a cryptic splice site. And they determine, you can determine what specific sequences upstream or downstream will determine the usage of this particular splice site. So this is the structure of the mini-gene, the simplified structure. And what we found was indeed using this mini-gene, we can determine that the mutants actually use the novel splice site way more than the wild type. And this is also shown here. And we looked at how exactly do they differ in their branch point use. And what we found was for the canonical branch points, which is a branch point where the adenosine is jutting out like this, and then there is base pairing between each of these, either wobble base pairing between G and T, or between the classical base pairing, we are not able to detect any differences between the wild type or the mutant. So this is a wild type and this is the mutant. But when we looked at the weaker or the non-canonical branch points, we found that almost always the mutant ended up using the non-canonical branch points better than the wild type. And that is shown here, especially at the -1 site. The mutants seem to be accommodating a change in the canonical base pairing requirement. So, and we did multiple other analysis, which I'm not going to go into the detail of, and we also determined the free energy distribution between these two data sets. And we came up with the model, which has been recently published, that in wild type spliceosomes, they use the canonical branch point and use the canonical 3' splice site. In the mutant case, they, for the most part, would use the canonical branch point, but it's also permissive on occasion to use a non-canonical branch point, which may be upstream. And that is the reason why you get some amount of missplicing. And that also explains why you don't always get 50% of the genes to be misspliced. So this is probably still favored in most of the cases, but also, there is a permissive nature of the mutant to use non-canonical branch points better than the wild type. So to wrap up soon, can aberrant splicing of single genes explain oncogenesis? This is one of the the big questions in the field. And multiple single gene models, some of them very extensive and high profile papers have been proposed, including recently BRD9, which is a non-canonical BAF component being misspliced by SF3B1, has been published by Omar and Rob Bradley's group. And previous reports on ABCB7, which is mitochondrial iron regulator, SLC25A37 or mitoferrin, which is also have been proposed. SRSF2 similarly, the initial papers have proposed EZH2 and DOTL1 being misspliced and hence contributing to oncogenesis, similar for U2F1. But there are multiple weaknesses for these models. And one is that the change in splicing has been relatively modest and not really consistent across the disease samples. And there are no common genes that have been shared between multiple cancers. And it is difficult to think that every single mutant splicing factor will actually end up missplicing one particular oncogene, and that is the dominant mechanism. So these have still not been widely accepted. Alternate mechanisms have been proposed, as I mentioned, that the mutant splicing factors lead to stabilization of R loops. So R loops is a phenomenon where during transcription, there is RNA coming off the transcriptomic machinery, but they form a triple helix in which the RNA and DNA is base pair, and if it is not removed immediately, that can lead to genomic instability. And 3 groups have found that there is increase in R loops in mutant splicing factor, but all of them have been overexpression models, so the relevance to real disease has not been demonstrated. U2F1 has also been shown to have altered protein translation, alternative polyadenylation site usage, and then we have collaborated with group in Georgetown about how SF3B1 interacts with cohesins and chromatin, and if this is altered in disease state. So a lot of work still ongoing and needs to be done. Finally, can we target this therapeutically? So early on in this in the spliceosome story, there was this concept of splicing vulnerability that was proposed, and companies started working on non-specific inhibition of the spliceosome. And this is not a new approach. And I think in early 2000s, there were phase 1 trials of using E7107, which is a palladianylide derived analog, which was dropped because of high toxicity. A newer modified version called H3B8800 was used by H3 Biosciences, and a phase 1 trial that recently concluded with fairly large 84 patients. They had no toxicities, but there were no PR or OR, no partial response or overall responses, so complete responses, so disappointing results. Other approaches, including targeting other spliceosome mediators like PRMT5 or ATR inhibitors, are also ongoing. So just to summarize, mutant splicing factors appear to be clearly oncogenic in specific tissue contexts, so not all cancers have these, some of them have it. biochemistry of splicing aberrations have been detailed using next-generation sequencing, and they don't seem to have much of overlapping changes between the multiple factors. The specific mechanism of oncogenesis is a very heavily studied area, and we are still not sure whether there is a single gene missplicing mechanism or global mechanisms that is not related to splicing that is leading to this. And targeting the splicing vulnerability, the initial study so far has been disappointing. So I'll stop there and acknowledge my lab, Abhishek, who has done most of the work, and Kiran, who has done the informatic work, past and present ones, and multiple collaborators within, especially Carla Nukubar in biochemistry. She and I collaborate heavily on this, and Stephanie, by virtue of working on SRSF2, we obviously end up in the same space a lot. And also my clinical colleagues who helped me with this endeavor while running a lab and then tolerating me and keeping me happy when I'm on service. So I'll stop there.
Thank you. Questions? David.
So this is a really hard problem. Doing all the right things from a molecular standpoint, but we're in a space where there are possibly 300 things, 250 of which are unimportant, 50 are important. So with these kinds of problems, sometimes genetics or biology are very helpful. On the biological side, perhaps is there a convergent process downstream that is the proximal cause of the hematopoietic dysregulation? So that's basically question one. And secondly, it seems like maybe this would be a case where when we want to look for an answer or suppressor gene dysregulation to CRISPR change or whatever.
Okay. So for the first one, yes, people have looked at pathways, and some pathways are always heuristic based and often forced. So we do find pathways like RNA biogenesis, and some of the signaling pathways do converge, but it's not really very convincing single pathways. It's multiple things are dysregulated, and we are also looking at least for the patient samples, really way past the initial stages of oncogenesis. So we really don't know whether any of those are relevant. The second question was about using screens. Yes, I think the BRD9 story came out of screens, although they used a mouse cell line BAF3 to use as to screen, where they themselves say those mutations don't actually happen in the mouse cell. So at least for SF3B1, it is really difficult because they use the branch point is typically not conserved. Exons are conserved, introns are not conserved between mouse and human. So how much of it is relevant? But screens have been tried, and some of these stories do come out of screens. Yes.
What do you think about the kinds of data on erythropoietin and SF3B1?
Yeah, I'm aware of it. I mean, it's obviously.
Just repeat it because
yes, the erythroferrone. And so erythroferrone being a regulator of heme uptake, I think, is they had linked it, and I think again the data is I don't see biological data that would actually convincingly show that dysregulating it. Just like the ECH2 or DOTL1, there is some probably genetic data, but not real functional data. Yes.
I think we probably should just.
Sorry.
Speaker. So,
thank you.
No, thank you.
I will answer questions later.
So turning